Okay guys, let us now proceed with this one. If I'm not mistaken, we ended on that portion about Western blot, so we are now in the last 18 slides for that of our hybridization, and of course, just an overview for our NAAT, just definitions. Okay, so we have here guys the topic which is known as fluorescence in situ hybridization. Of course, by the term fluorescence, we're making use of a certain fluorescing substance, of course. And for the information of everyone, guys, it is somehow still applying the principle of hybridization. So there is still base pairing. So that specific process of a certain complementary base pairing is still present. But this time, guys, it is differentiated from that of northern blot, from that of western blot, and of course from that of southern blot because it is in situ. In situ means, okay, let's say for example, in an original place, of course, site, or of course location. In an original place, site, or of course location. That's why it is different from that of the others because we are making use of intact cells here. So the cells remain intact. There is no need for us to lyse the cells. There is no need for us to lyse those specific types of substances from which we are supposed to get the DNA from or the RNA from. But rather, the cells will remain intact. So there is no need for lysis of cells, and you will be able to see hybridization happens inside the cell. So that specific probe that we are using here must again be able to pass. This probe passes through the cell membrane. Okay, please take note of this one. This probe passes through the cell membrane. So there is passage through that of the cell membrane. So therefore, guys, the cell remains intact and the probe will just hybridize inside the cell, specifically inside that nucleus. So that is for fluorescence in situ hybridization. Of course, we're going to make use of fluorescein here. Usually, C, the most common, fluorescein isothiocyanate as one of the most common fluorescing agents utilized for hybridization. So they usually make use of FITC, or fluorescein, isothiocyanate, fluorescein isothiocyanate. So please take note, that is for our fluorescence in situ hybridization. So what about this specific type of testing? Okay, so fluorescence in situ hybridization, or FISH, is a molecular biology technique that can be used to detect microorganisms known to that of biodegradable contaminants, or which means, guys, those specific types of microorganisms that can biodegrade contaminants. So it was even stated or proposed that it is very much applicable to bioremediation. Let's say, for example, guys, when we speak of bioremediation, we have here the treatment of different facilities, water, and of course biosystems with a certain agent that will somehow clean that specific biosystem from the contaminant and of course prevent more pollution from somehow affecting different biosystems. So this is very much important in industries before. But as of this moment, guys, I would like to point out that it has that specific relevance now in medicine, specifically under diagnosis. Next one, guys, it's also for monitoring, and of course for targeted treatment. And of course for targeted treatment. It is for diagnosis, for monitoring, and of course, guys, for targeted treatment. That is for our fluorescence in situ hybridization application. But before, it is mostly applied for that of bioremediation. Okay, so let's continue. When combined with traditional measuring of changes in contaminant concentration over time, FISH provides project managers valuable information for site management, including site conceptual model development, remedy selection and optimization, and determination of contaminant attenuation rates. This is just, again, more of industries. Okay, so this definition here is associated with maintaining a specific program that will monitor the spread of pollution and of course counter that one in order to maintain that specific status, which is known as, yes, if you are to remember, guys, sustainable development. Remember, as we grow when it comes to that of our industries, the more also is the effect in our environment. That's why they propose sustainable development in order for us to have these increases in our industries in any other advent of technology without sacrificing that specific, let's say, for example, environment itself, or of course, all other ecosystems. But for this one, guys, it's mostly in industry, but the one that we gave here on top, or I gave here on top, is actually the purpose for our discussion today. Okay, so let's continue. So the question that we have here is this: how does it work? How is it possible that a certain type of probe can enter the cell and of course can hybridize with a substance inside the cell? So this is what usually happens. Short sequences of single-stranded nucleic acids, which are of course DNA, called as gene probes, are designed to match a portion of a gene or metabolic product of the organism or the population of interest. For us here, let's say for example, this is for cancer diagnosis. Okay? So we're going to look for that specific type of sequence that is of course somehow showing a cancer, let's say, a cancer relatedness, or of course probabilities for cancer due to that mutation. So let's say we have here ATA, CC, or CG, CG, okay? This is our, once again, gene of interest. Of course, just the same with our blotting techniques, we need to create a probe. And this probe must match our own sequence for GI. So let's say, for example, our probe here must be one, how much, this, or how many, 1, 2, 3, 4, 5, 6, 7, 1, 2, 3, 4, 5, 6, 7. So we have here T, A, T, G, C, G, C. So the probe must match that specific gene of interest or of course DNA fragment that we are looking for. And of course, for some other applications, population of interest or microorganisms under bioremediation. Next one. A fluorescent dye is attached, that's why it's fluorescence, okay, because it is fluorescent dye. A fluorescent dye is attached to the probe so that when the probe binds to the target sequences within the cell, take note, it has a fluor with it. Okay, guys, it emits fluorescent light that can be observed through microscopes. Of course, using epifluorescent microscope or sorted with flow cytometry. These are actually the two major methods. Firstly, microscopy, or we can also make use of flow cytometry. This is on cell counts. So guys, because it must hybridize inside the cell, you are to expect that this probe can easily cross, okay, can easily cross membranes, because this probe is also somehow powered by, please take note, lipid layers, okay? So it has lipid layers, which means it is fat soluble. Because it is fat soluble, remember guys, your lipid bilayer here is a phospholipid bilayer. Your lipid layer here is a phospholipid bilayer. Because it's a phospholipid bilayer, the probe that is labeled with a fluor can easily pass through membranes. Remember, all of our organelles and even guys the nuclear membrane and of course guys the cell membrane itself is made up of a phospholipid bilayer, which is considered as, if you are to recall, which is considered as an example of, yes, a fat soluble substance. And because we make use of a certain probe that is also somehow governed and of course covered or encumbered with fat soluble substances, it is able to enter the cell without once again altering the characteristic of the probe itself. And the probe also carries a specific type of label, and this label is a fluor. Okay, so what happens here? If this is our gene of interest, the probe is able to enter and it hybridizes with that specific gene of interest below. And of course, guys, we can see that there is a fluor attached to it. And upon exposure to that of radiation, it can be seen through microscopy or can be counted under that specific process known as flow cytometry that we will discuss as we progress. Okay, because it's recorded, you can pause, you can again replay to have a complete grasp of the necessary information, and you have screenshot mechanisms. Next one. Guys, let us now continue with the next part of this line. Okay, so cells emitting a fluorescent light are called as hybridized cell. Of course, if they emit a fluorescent light, it means that within that cell, a certain hybridization happened. and the fluor is again present as a tag. So those cells with hybridized, okay, gene of interest are referred to as hybridized cells because of the fact that they contain that specific hybrid that contains the fluor, which is exposed to that of your radiation and will emit a fluorescent light, thereby making it again easier for us, the microscopists, to see whatever is supposed to be seen. Next one. In flow cytometry, labeled cells are diluted or concentrated depending on the initial cell concentration in the sample. So that individual cells pass through a laser beam that detects and counts fluorescent labeled cells. Wait lang yun guys ha. I'm just going to check my message ako. Basta iba lang important. Wait lang yun for the meantime. Okay, so kindly take note of this one, guys, as what was stated here, what will happen. So as you can see, guys, in this case, guys, in flow cytometry, the cells that are labeled are diluted or concentrated depending on the initial cell concentration. So that individual cells pass through a laser beam that detects, that counts or counts your fluorescently labeled cells. So let's say, for example, we have here the laser beam. As the cells pass by, they will be counted. If this one contains the fluor or if this one does not contain the fluor. So which means, guys, if it contains the fluor, there will be, once again, that specific type of counting that, yes, this is indeed a hybridized cell because it emitted the fluor, it emitted fluorescence, therefore it is counted as one of the hybridized cells. Flow cytometry can be significantly more efficient than counting cells using a microscope. Of course, no, counting cells using a microscope with that of the premise of fluorescence may be subjective at times. So when we speak of subjective, it is again prone to errors by that of a certain person doing the test or by that of that specific medtech who is assigned to do that specific microscopy. Okay, so flow cytometry is usually standardized because we're making use of controls here. Next one. Cells in environmental samples are handled in such a way that the cell structure remains intact while still allowing the comparatively large gene probe to enter the cell and bind to the target gene within the microorganism of interest. So take note of this one, guys. They will be handled in such a way that the cell structure remains intact, again intact. So this will still allow comparatively large gene probe to enter because of, again, fat solubility and bind to that of the gene of the microorganism of interest or to that of the gene of interest because we're making use of medicine here. Under ideal conditions, only cells that contain the target gene are recognized by the probe and become fluorescently labeled. So again, the probe is very specific. It will not label or it will not once again bind with any other sequences that is not again considered as able to base pair or hybridize with it. Various cell staining procedures are sometimes combined with fish probes to allow quantification of various parameters such as total number of microorganisms or the presence of specific compounds. So take note of this one, guys. This is the usual when it comes to that of the reaction itself. Okay, let us now continue with that of the next one. Okay, so guys, as you can see in this picture, we have here our samples. From that of our samples, guys, what happens here is that the probe will again bind with that of any other gene of interest that is present here. But the probe are usually labeled with our, again, fluorescence. So what will happen here? In the presence of the gene of interest, it will bind with that of the gene of interest that will be determined as a certain, again, emission of fluor or that microscopic presence of fluor when it comes to that of either, please take note, flow cytometry or microscopy respectively. So in this case, guys, those cells may be seen as hybridized through passing to a certain laser beam with the use of flow cytometry or through that of our epifluorescent microscopy, through that of your microscopic detection, which is subjective, or flow cytometry, which is objective. Okay, guys, let us now continue with the next one. These are examples of selected fish probes or cellular stains. For the information of everyone, you are not anymore required to kindly spell them out. All you need to do is to memorize if they are once again utilized for such purpose, or what are the characteristics. The first one is the DAPI. Guys, no type of contaminant present when it comes to the testing, so which means it's very specific. DNA of all microorganisms, dead or alive, are still somehow considered as probable targets for this specific type of stain. Next one, guys, reference. This is a very common laboratory cellular stain and not unique to environmental contaminants. Next one, acridine orange. Also very, very famous siya. It is able to, once again, guys, target all DNA of microorganisms, both live and, of course, dead microorganisms. This is a very common laboratory cellular stain also, but not unique to environmental contaminants. We also have here, guys, the DE, okay, the DE 12590 and, of course, 1259C, which are contaminated by that of chlorinated solvents. For the information of everyone, some Dehalococcoides species for that of 16S rRNA are target microorganisms. Reference was taken from Young and Zayer of 2003. Next one, KT1Fe, which is again contaminated by that of trichloroethane for that of target microorganism Ralstonia eutropia. If you are to remember, guys, these are all for bioremediation man lang. Selected fish probes example lang na siya. Next one, AC627BR, naphthalene is the contaminant, and this is for naphthalene deoxygenase or dioxygenase, which is again referenced by Baker, Mans, and Madsen in 2002. So these are all for bioremediation. And RH-Lu, S-triazine herbicides, Rhodococcus ratislaviensis, which is, of course, 16S rRNA from Gray et al. 2009. So these are just examples of our selected probes or fish probes or cellular stains. Very easy man lang, guys, no, ang aton nga fish. So how are this data reported, or how are the data reported? For the information of everyone, depending on the method, fish results can be presented in two different ways. Again, depending on the method, fish results can be presented in two different ways. If fish is evaluated using microscope and manual counting of labeled cells, the results are presented as cells per unit, which is again somehow again liter of liquid or gram of solid. So if fish is evaluated using your epifluorescent microscope and, of course, manual counting, which is again very subjected, with the use of, again, labeling, remember, we are supposed to look for the presence of labels, guys, the results are presented as cells per unit. So number of cells per unit. Let's say, for example, per UL, per ml, per liter, or, guys, if it is, again, per gram of solid. But most of the times, per unit of liquid or per gram of solid. So that is for microscopy. But if fish is evaluated with advanced microscopy technique or microscopy techniques and digital imaging processing, the results are usually presented on a relative volume or area basis, which can also be converted by the laboratory to cells per unit of liquid or solid. So usually relative volume or area basis. So it can be volume by area, or, of course, guys, we are talking about the usual counts with the use of automated machines. Okay, let's continue. What are the different advantages for that of our fluorescent in situ hybridization? Number one, fish does not require cultivation of the organisms or any technology-based gene amplification, which can lead to false negatives and false positives. Bongga siya, guys. It does not require cultivation of organisms, which means one thing, no need for gene amplification, no need for isolation, no need for extraction, because it can be determined while the cell is still intact. No need for further isolation and extraction techniques. In contrast to some other methods, fish allows visualization of whole cells that are important to environmental remediation activities. So again, guys, it is for remediation, it is best for remediation. Fish can thus provide complementary information such as morphology of the cells or association of groups of microorganisms with relationship to one another. It is because, as you can see, the cell here is intact, and because the cells are intact, they are again seen as to morphology. They can be somehow explained as to that of your specific type of, let's say, for example, ng structural or cultural characteristics such as for microorganisms, and everything can be somehow seen as is. Next one, guys, fish can target several different genes simultaneously. For example, genes associated with specific degrading species of interest such as Dehalococcoides and broader microbial groups such as methane-producing organisms. This one will somehow differentiate it from that of our previous types of blotting techniques for southern, northern, and of course western. Why? They are differentiated because, as what was stated here, they may target several different genes simultaneously. For the previous one, only one gene of interest. For this one, we can have several gene of interests in just one specific cell. So which means, guys, we can just make use of different colors or different labels or different fluorescence or fluoresceins. Next one, depending on the species and in combination with other appropriately validated activity-targeted approaches, fish can provide general information about the activity of organisms or populations of interest. So it is not only for that of the gene of interest, it can also be for the biochemical characteristic of the cells in question. So this can be also considered as a higher level technology for microbiology, specifically not only for cultural characteristics, but also for the characteristics of cells as they are again grouped or as they are placed in a certain environment. Fish enables single-cell microbial studies and allows for subsequent studies such as the one that we will discuss in the future, gene sequencing. Guys, sa gene sequencing. You will just see that in gene sequencing, it's very important for us to somehow identify the sequence that may be correlated with the disease and the sequence which may be beneficial to us. You will just know that one as we progress. But when it comes to that of fish, it also has a certain limitation. For the information of everyone, the detection limit of fish is high. It is up to 10 raised to the power of 6 cells per ml. However, in some cases, high detection limits can be corrected by sample concentration or cell extraction methods which lower the detection limits to a few hundred cells per concentrated samples. So when it comes to that of high detection limits, the higher the detection limit, the probability of also producing, take note guys producing a specific false positive result. So we can also somehow limit this type of detection limit while this specific process is said to be undergoing, or of course this specific process is ensuing. We can still make use of some extraction mechanisms, or of course we can make use of isolation mechanisms that will somehow lower the detection limit so that it will be the actual gene of interest or the actual cell that we are looking for. So again, limitation is very high. It is 10 raised to the power of 6. It's also very similar with a very high sensitivity value. The greater the sensitivity value, that specific detection is said to be considered as very prone to false positives. But the smaller or the lower is the sensitivity value, I mean the number for sensitivity. When we speak of the sensitivity value, we are talking about the number for sensitivity. The higher the number for sensitivity, please take note, the lower is that, the higher the value for sensitivity, the higher is the false positives. Next one, the lower the value for sensitivity, guys, the lower that specific number for sensitivity, please take note, guys, the lower is the probability of false positives, because we're talking about positives when it comes to that of sensitivity. When we speak of specificity, we're talking about negatives. Again, the higher the number for sensitivity, take note, isulat ko na lang, the higher the number for sensitivity, the greater is the false positive probability. But the lower the number, okay, dugangan na lang sa unahan, okay, because we're talking about comparison. The lower the number of sensitivity, we're not talking about that specific interpretation. We're talking about the number. The lower the number of sensitivity, the lesser is false positivity. Okay, so take note of that information. So, hindi magalit pwede, no, na super taas gid ang iyang sensitivity. It's not 106, it is 10 raised to the power of 6. Please correct that one. Okay, let's continue. Validated probes and fish process or procedures rather, again, validated probes and of course FISH procedures are not available for a wide range of organisms within the bioremediation field. But again, it is now applicable to cancers such as, it is now applicable to cancers such as breast cancer and of course guys, ovarian cancer and cervical cancer. So it's not applicable to breast, ovarian, and cervical cancer. Additionally, standard protocols for sample collection and storage prior to FISH analysis have not yet been developed. But if I'm not mistaken, for microbiology, it is already standardized. And of course, for these tumor markers in breast, ovarian, and cervical tumors or cancers. So tumor markers are now being measured with the presence of FISH itself. FISH can also be used to target not only ribosomal genes, which indicate the type of organism, but also functional genes via mRNA relevant in bioremediation. These other genes indicate what the microorganisms can do with regards to contaminant biodegradation. For example, naphthalene dioxygenase or reductive dehalogenase. However, laboratory protocols are often time-consuming and complicated and not validated for field applications. Okay, for the information of everyone, when it comes to that of contaminant biodegradation, guys, there may be some contaminants that may stay all throughout even if they have been treated. So it's very difficult for us to somehow, again, validate which is again somehow able to stay for a longer time, or of course if the half-life is longer for that specific type of substance. But again guys, this is mostly in microorganisms, but when it comes to that of tumor markers, they are already established. The FISH method is not widely used commercially, okay, or it is not widely commercially available. Currently, many specialized research laboratories are performing this analysis to explore and optimize the potential of FISH for validated and cost-effective studies. But always remember, its main, again, difference is that we are examining the presence of that fluorescein labeled probe inside your cell while it is still, please take note of that information, while it is still inside the cell. Next one, guys, this is the last topic actually for our nucleic acid amplification test. Guys, if you are to remember, we're talking about nucleic acids, so it can be DNA or it can be RNA, and of course amplification, increase in numbers. Amplification, increase in numbers. Guys, in our nucleic acid amplification test or NAAT, this is a type of diagnostic test for the presence of nucleic acids that are amplified for detection. So NAATs detect genetic material or nucleic acids and sequences that comprise the genetic material being searched for. Let's say, for example, we have here the gene of interest. So this is starting from 1. This will become 1 million, and so on and so forth. Okay? Remember that information. So when it comes to that of NAAT procedure, if you are to remember, the best example is PCR. It works by first amplifying or making many copies of the genetic material, if any is present in the person's specimen. So if it's present, you're going to somehow multiply it, isolate, extract, multiply, and of course, it will undergo several cycles until we reach the number that we are looking for. Amplifying those nucleic acid enables NAATs to detect very small amounts of DNA or RNA in a specimen, making this test highly sensitive for diagnosing viral infections, of course, for the diagnosis of COVID-19. In other words, NAATs can reliably detect small amounts of nucleic acids and are unlikely to return a false negative result of the infectious disease in question. So guys, which is again telling us that your NAAT here, including PCR, is very, very sensitive and of course specific. The lower the sensitivity number, please take note, the more specific it is. So guys, what about this one? Okay, so let's have here the different examples of NAATs. Actually, NAATs can use many different methods to amplify nucleic acids and detect the virus, including but not limited to these two most common. Firstly, reverse transcription PCR or RT-PCR, the one that we will discuss in the next few meetings. And of course, guys, together with that, our isothermal amplification, including, guys, the following. Ari lang i-memorize nyo ang ilalang meaning. Near, nicking endonuclease amplification reaction. Next one, TMA, transcription mediated amplification. These are actually board exam questions last time. Loop mediated isothermal amplification, also known as LAMP. Helicase dependent amplification, also known as HDA. CRISPR, also referred to as clustered regularly interspaced short palindromic repeats, and of course SDA or strand displacement amplification. All of this will be discussed as amplification techniques in our next meeting. Okay? So that ends our prelim topics. Actually, na-cover din natin ato na coverage. Only that, guys, no, we are again lacking time as to that of, yes, the holidays. That's the reason why next time we're gonna make use now of some laboratory sessions. Okay? Thank you very much for bearing with me, and God bless you on your exam. God bless and take care.